And now the fun stuff begins, because we get to start to crack that code, having the order of the letters in front of us. But fundamentally, it is very much a digital code.
The secret of life itself, the DNA molecule, a genetic discovery that could give man the ability to create life to specifications. With it comes the power to change evolution itself. Never have we had such opportunity, or such awesome responsibility.
Change DNA structure in the lab is fairly straightforward and it's fairly simple. You can study a piece of DNA to unravel its function. You modify the composition and you see what the effect is on function. That's daily routine in a lab that has this technology. Going from the lab to the person to therapy, in theory, is the same thing and is as simple. In practice, it has been shown that it's a lot more difficult. And the reason why it's so difficult is not that we don't know what pieces of DNA to put in, is that we do not succeed sufficiently and at a high rate in getting these pieces of DNA in the right cells. But we have learned how to do it from nature. What is a viral infection? It's a virus that sticks to the cells, that enters the cells, and that uses the machinery of the cells to multiply itself. Now you take this virus, you cut out the pieces that are dangerous so that it will still stick and be engulfed but not develop in the cell. And then you add to this some other tools that the virus then can use to cut out the bad DNA, stick in, splice in the good DNA. This is a mechanism that is used now to do gene therapy using viruses.
The ability to tamper with our genes has obvious applications. We can change DNA to prevent or cure hereditary diseases. We can alter the DNA of donor organs that are to be used for transplants so that the body of the recipient won't reject them. The organ doesn't even need to come from a human being. It could perfectly well be that of an animal.
Pigs would be a good supply of kidneys or other organs to transplant in humans. But because of the difference between the two species, the pig kidney or the pig liver will be rejected by the human. So if we can humanize the pig kidney by putting in some human DNA, which will make the cells look a little bit more human, so that they are not rejected by the person immediately, then there's a better chance that you'll have a take. And this is tried to humanize animal tissues, and it's becoming a big industry because we need more organs.
I think increasingly we're going to start realizing that this body is not sacred. The way we are is not some kind of God-given plan. It's really a pure random accident. We take two sets of genes and we shuffle them and something comes out. Sometimes it's a wonderful product, sometimes it has a hole in the heart, sometimes it has psychosis or tendencies towards extreme anger, has addictive problems, can't concentrate, all kinds of defects. To say, oh, that's normal, that's sacred, that's good, to me is rather absurd. It's random. There's not a plan there that we're thwarting. So genetic engineering seems to me one of the most moral things we can do.
A single species is defined by the isolation of our genetics, reproductive isolation. But when you begin to take genes from different species to mix together all of the genetics that is available among all animals, and also to design new alterations to the genetics, well, reproductive isolation really doesn't have much meaning anymore. And so you can imagine all sorts of forms and that will evolve in the future.
Once we know exactly how our DNA code functions, we will be able to enhance our bodies. Human life will improve from one generation to the next as we control and accelerate natural evolution. But a rival to mankind is appearing on the horizon, a rival that is evolving much faster.